So tonight we don't have the TV, so we can do a more informal introduction. I'm Tom Zinnan. Thanks for coming here. I work for the Biotech Center. I also work for UW Extension Cooperative Extension. Wednesday Night Lab is sponsored by Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance. So thanks again. We do this every Wednesday night, 50 times a year. Now I get to ask you 5 questions. You have not been prepared for this.
I have not been prepared for this, yes.
You are prepared?
No, I have not.
You have not been, see? Testimonial. This is great. Are you now or have you? Never mind. Okay. Where were you born?
I was born in Chennai, India.
Chennai?
Yeah.
C-H-E-N-A-I?
C-H-E-N-N-A-I, yeah. Though I've never lived there, I was just born there, and then the rest of my life I spent in Bombay, so Mumbai, India.
Okay.
And then that was until undergrad. Yeah.
Until undergrad.
Then I left home, and then I never went back.
Where'd you go for undergrad?
I studied the National Institute of Technology, so that's that's in the south of India. So
okay.
It's in a city called Mangalore. It's very beautiful out there on the coast.
Yes.
And that's sort of where, living, all my life in the coast in Bombay. Then I went to school in Mangalore, and then, grad school again, studying the oceans. So that's sort of, I've maintained a continuous link to the oceans.
Well, I grew up in Dixon. That's true. And where'd you go for your PhD?
Michigan. So this weekend was interesting. I did turn the TV off once it was 21-0. Okay.
What was the final score? By the way, that was you've heard of the ontological proof for the existence of God. Last Saturday was the football proof for the existence of God. That was amazing. And when did you arrive here?
In 2007, so I've been in the US for 12 years now.
Great. So you're at the Department of Bacteriology?
Yes.
Very good. And I don't know if you had a chance to read my little missive, but viruses are one of my favorite topics. I've never heard of ocean viruses. And so once I first saw the stories on your work, it was like, whoa. I'm looking forward to hearing you talk about this work, shining a light on dark ocean viruses. Please welcome, I don't know how to pronounce your name for sure, Karthik Anantharaman?
Yep, that's good. Yeah. Thanks for that lovely introduction, Tom. Can everyone hear me in the back all right? Okay, that's great.
A little bit.
Little bit, okay. Now?
Great.
Yep.
Thank you.
Fantastic, yeah, that's great. So thanks a lot for this invitation. I'm very happy to come here and talk to all of you. And today I'm going to talk to you about viruses in the oceans. And when I say the oceans, I'm primarily going to talk to you about viruses in the very deep depths of the oceans. Viruses that inhabit hydrothermal vents, which are these deep sea volcanoes. Very, very interesting ecosystems in their own right. And then what hopefully I can do is give you a sense that there's a lot to learn from viruses in the deep oceans, and then actually apply that into our lives, let's say into our understanding of human diseases, or even our understanding of ecosystems in other places. For example, we can take a lot of things from the deep oceans, apply that to Lake Mendota, so freshwater ecosystem. So there's a lot that we can learn from studying one ecosystem and actually applying that. So hopefully I can give you a sense for that, and I'll show you a lot of interesting examples. Through the course of my talk, I'm also going to show you a lot of pictures and videos. Since we do work in the oceans, we do a lot of field work, so that implies going out on boats. Not boats, these are big ships, but yeah, it'd be problematic if we went down on a boat. But we do go on these big ships, and then we use submarines, remotely operated vehicles, robots to sample the oceans. So I've been down in submarines down to the depths. So hopefully, you can appreciate some of the field work that we do. And I have some videos and pictures as well. So let's begin, and I'll just tell you a little bit about the lab that I run. I've been here for about a year and a half now, and I run what I think is a very modern microbiome laboratory. So we work at the interface of 3 different fields. One is biogeochemistry or chemistry primarily. This is very geoscience focused, so more geology focused, let's say. We do a lot of computational biology because when we study organisms in the oceans, we often can't bring them up. We can't really see the viruses, you can't bring them up, and a lot of times you can't really culture these microorganisms or viruses in the laboratory. So how do you study them? So we have to use DNA or RNA sequences. So it's primarily sequencing based, and then everything is computational biology from there on. So we develop our own algorithms, our own softwares to analyze all of this. And finally, my bread and butter really is microbiology and microbial ecology. So everything that comes from culturing, as well as, so culturing is growing organisms in the lab, or studying the DNA sequences. We pretty much do all of it. Now, as far as my lab is concerned, right now we have 3 very different themes that we're running in the lab. So we study hydrothermal vents in the deep oceans, and here we primarily study inorganic sulfur. So I forgot to mention, but everything that our lab does is associated with the element sulfur. Sulfur is an extremely important element on Earth in general, and I'll talk about sulfur as we go further in the talk. But we study hydrothermal vents and the deep oceans, and here we study a lot of inorganic sulfur and how microbes and viruses actually use inorganic sulfur. We've got a lot of field sites around the world. So we have a field site at East Pacific Rise in the Pacific Ocean. So this is off the coast of Mexico and Costa Rica. Sware is close to the Maldives in the Indian Ocean. Actually, I should just fix this. I forgot to close my Slack channel and it's just someone messaging me. Apologies, but just 1 second, else this is just gonna keep popping. Okay, so to just pick up where we left off, we've got a lot of field sites. So we study hydrothermal vents around the planet pretty much. So we have a field site, East Pacific Rise, that's just off of North America. SWIR stands for the Southwest Indian Ridge, so this is in the Indian Ocean. Lao Basin is right between the Kingdom of Tonga and Samoa, so that's close to Fiji, so really out there in the Western Pacific Ocean. And then finally, Guaymas Basin was the one I visited most recently in March of this year. And this is in the Baja Peninsula. So the Baja Peninsula is rifting, so it's moving away. So there's a lot of hydrothermal vents in Baja Peninsula. And my estimate, or say in my opinion, this is probably the most interesting site amongst all of them. It's just a really, really beautiful environment down there and a very, very interesting ecosystem. We also work in the human system, so human microbiomes, the gut microbiome associated with IBD, which stands for intestinal bowel disorders, colitis and everything associated with it, and colon cancer. And my lab has also just started working on freshwater microbiology of very deep lakes. So this is Lake Tanganyika in Africa. And Lake Tanganyika is actually a very interesting system because it turns out, we didn't know this when we were going in, but it turns out that it's very similar to the oceans. It's very deep. Large parts of it have no oxygen, and it's also got hydrothermal vents. A lake with a hydrothermal vent, how cool is that, right? So why do we study hydrothermal vents? Hydrothermal vents can be really, really important for ocean chemistry as a whole. So they act as an interface between the Earth's crust and the oceans. So the Earth's crust is spewing out material all the time. Whatever's below the crust is pretty hot, but the crust itself, as you go beneath the surface, it's very hot. So as the water goes in, it heats up, it picks up a lot of metals, a lot of elements, and then it spews them out in the ocean. This is a really important process. To give you a sense for it, here is longitude, so you can get a sense for the large amount of area we're talking about. And here's a hydrothermal vent right here. And what you're seeing are ion concentrations. So the warmer colors show higher ion concentration. So what this is showing is that when you have a hydrothermal vent, the ion concentrations around it are very high, and then slowly, it goes away. But the important takeaway message over here is that wherever you have a hydrothermal vent, whatever elements come out of it, don't just, the concentrations are not just elevated around it, they disperse throughout the ocean. And that's really important, because when you have these plankton or say algae on the surface of the ocean that are fixing carbon for us, let's say, these organisms, where do they get their iron from? About 50% of their iron budget is actually coming from hydrothermal vents that are sitting 3,000 meters beneath them. So that's the importance of the hydrothermal vents. This is again a very similar figure. So this is showing the entire world. And what this is showing is the total concentrations of iron and manganese. So the darker colors show elevated concentrations. So I just mentioned our field sites are around here, a couple of them. And you can see there are hydrothermal vents all along, and then there's elevated concentrations right where the hydrothermal vents are, but the concentrations are elevated pretty much throughout the oceans because of them. So these vents, as I just mentioned, are extremely important sources of iron and manganese, and then they affect ocean chemistry on global scales. But why do we study sulfur? We study sulfur because it turns out that it's sulfur that actually controls how much iron actually gets out. So the concentration of sulfur is critical to the concentration of other elements and the solubility of other elements in the water. Now, when we think of, say, hydrothermal vents, they are also major sources of organic carbon. Where does the organic carbon come from? It comes from microorganisms and viruses. Microorganisms and viruses take inorganic carbon, which is carbon dioxide. The same carbon dioxide that's in air is also in the oceans. Microorganisms and viruses actually have a lot of organic carbon, and that gets transferred to the deep oceans. Hydrothermal vents also disperse elements, microbes, viruses, and larvae. So the dispersal happens because a lot of these fluids are really warm. So when you push warm fluids into a cold environment, there's a density difference, they want to rise up. That's how dispersal happens. And they actually, because of these temperature gradients, there's a lot of biogeochemical transformations. Biogeochemistry just means, it's a very complex interplay between biology, geology, and chemistry. A neat dance is the way I'd like to think about it. Now, when we think of hydrothermal vents, are they localized? Are they globally distributed? How widespread are they? If you think of the world as a baseball and you think of the seam, that's exactly how hydrothermal vents would look. So these lines are what are called mid-ocean ridges. So there are these large mountains at the bottom of the ocean, and they basically crisscross our entire planet. Wherever you go, you're not too far away from a hydrothermal vent. Every single ocean, every single basin has hydrothermal vents, and clearly we haven't visited all of them. So there is a lot of hydrothermal vents out there that remain to be discovered. But we've got a lot of field sites. We've got field sites. So this is Guaymas Basin, right here in the Gulf of California. Lau Basin's here. This is East Pacific Rise. And then finally, Southwest Indian Ranges out here. So now what I'd like to do is just show you a video to give you a sense for how beautiful these ecosystems are. Hydrothermal vents are not just a volcano spewing out hot liquids. And it's not just small organisms that live there. There are large organisms. So in this video, you'll get a sense for how beautiful this ecosystem is. And I'll also pause and show you what some of these colors actually mean. Okay, let's take a look. Oh, so I must mention, so this, before I start this video, this actually came from the most recent cruise that I did. So this is this Schmidt Ocean Institute on their ship, and this was in March of this year. And for the first time, a lot of the videos I'm going to show today are all entirely in 4K, so the highest resolution possible. So we're actually using these cameras in the deep sea. So a lot of the footage and the resolution you're going to see is going to be pretty unparalleled.
Oh my God.
Isn't that amazing? Primordial juice, fueling all of the biology that you see around the system.
So yeah, all expeditions like this are highly collaborative, you know, so there's a lot of scientists that go out together. So was the lighting okay? Could everyone in the back see the video pretty clearly? Okay, so I don't need to turn anything up. Okay, that's great. So all of the colors that you actually saw in here, you know, there's a lot of these colors, white, yellow, orange, all of those are actually biofilms that are entirely made of microorganisms, mostly bacteria. And nearly all of those bacteria are actually consuming sulfur. Specifically, they're consuming hydrogen sulfide. Hydrogen sulfide is a pretty toxic compound. We produce it in our gut, but in our gut it's pretty bad for us. It causes inflammation. It drives colitis, IBD, colon cancer, a lot of diseases. But these organisms are pretty happy to live in this toxic fluid, consume all of that hydrogen sulfide, and also look beautiful at the same time. So all of the macrobiology, so a lot of larger animals, they are also dependent on the hydrogen sulfide, on the bacteria that are consuming the hydrogen sulfide. And I'll show something further down the road about that. Oops. Okay. So how does a lot of this hydrogen sulfide actually get consumed? It actually gets consumed by a process known as chemosynthesis. This is very much similar to photosynthesis, except that instead of light, it's actually chemicals that are actually fueling this. So chemosynthesis is actually also referred to as chemolithoautotrophy. And essentially it involves carbon fixation using energy that's derived from reactions that involve inorganic chemicals. What does this mean? So chemo stands for energy source. So these are chemical reactions instead of light. So chemo instead of photo. And litho stands for the electron donor, the inorganic chemicals that are donating electrons to produce energy. Auto stands for carbon source, which is CO2, and all the CO2 comes straight out of the hydrothermal vent. And then finally, troph just means eater. So a microorganism, unlike us, can actually be very, very fluid with regard to their food intakes and what they breathe. So we can only consume organic carbon, carbohydrates, proteins, fats, they're all just, organic carbon. We can only breathe oxygen. But the microorganisms are extremely flexible. So they can consume hydrogen gas, hydrogen sulfide, methane. These are some of their food sources, let's say, in a hydrothermal vent. But in addition to breathing oxygen, they can breathe nitrate as well as solid minerals. So they can breathe iron oxides, manganese oxides. So when they're actually consuming the hydrogen sulfide, they can couple it to any number of reactions. That's why you get a lot of colors, you get a lot of diversity at hydrothermal vents. The sulfur chemistry in hydrothermal vents is you can say it's quite complicated and connected to a lot of different things. The critical thing is that sulfate really is the primary sulfur species in the environment. There was a time on Earth where hydrogen sulfide, the Earth was a very reducing environment, but now it's a very oxidizing environment because there's a lot of oxygen. But when the Earth was a reducing environment, the oceans were full of hydrogen sulfide. But today the oceans are full of sulfate. So there's a lot of sulfate in the oceans. The oceans are super saturated. And it's that sulfate in hydrothermal vents that gets completely transformed to toxic hydrogen sulfide. Sulfate reduction that happens beneath hydrothermal vents can drive the anaerobic transformations of carbon. So carbon, being organic carbon, gets pushed out as CO2. And when you think of, say, sulfate reduction, it's a pretty ancient process, about 3.5 billion years old. It's believed to be probably one of the earliest metabolisms that came about in all of biology, sulfate reduction and the production of hydrogen sulfide. When hydrogen sulfide is produced, it can get oxidized to sulfate. So this happens very, very commonly in hydrothermal vents. But what also happens is hydrogen sulfide forming organosulfur compounds. The reason I'm mentioning this specifically is that now we're going to start talking about viruses in a little bit, and viruses are very tricky. But specifically what my lab is interested in is not how viruses infect bacteria or other organisms and kill them. That's pretty straightforward. That's what viruses do, similar to predators. What we are specifically interested in is how viruses manipulate these organisms. So viruses can infect them, they can manipulate the organisms, and before they kill them, they can do a lot of metabolic transformations. That's what we are interested in, because when they're manipulating the organisms, what we assume to be, let's say, the chemistry of an environment would be completely different to what is actually happening. So we'll get into some specific examples associated with that. Now, when we are thinking about hydrothermal microbes, it's really important to know where you are in the system. Deep down there, let's say in the background deep ocean, there's not much around. So the organisms are there, but they're growing very, very slowly. But we can go in and go close to hydrothermal vents where there's a lot of hydrogen sulfide, and then we find that the organisms grow very quickly. And then you go away, closer, high up in the plume of the hydrothermal vent, and they grow, about medium to medium high. And all of this, we do it computationally. So again, developing algorithms to figure out how fast or how slow an organism can actually grow. Completely fueled again by sulfur concentrations. Now, I also mentioned that sulfur is very important to the animals. So I'd like to show a video that shows how important hydrogen sulfide is to tube worms. And when you think of tube worms, bottom of the ocean, never seen light, and we think plants grow pretty fast. But it turns out that these tube worms that I'm about to show you are actually the fastest growing organisms on the planet. Okay.
They have a lifestyle that is completely foreign to what we think of as animals. They're really cool. They rely completely on their symbionts for nutrition. An analogy would be, instead of eating, we would go out onto the street when we're hungry and sit in the sun to feed our bacteria.
Tube worms have a relatively high rate of uptake of hydrogen sulfide. The surrounding water then, if they pick it up, is less toxic for all the other animals. You can think as tube worms very similar to coral reefs. But in contrast to a bare rock, you have suddenly a three-dimensional structure with a lot of space to live.
The fact that these symbionts are so similar but occupy different environments speaks to maybe a metabolic flexibility of the symbionts, and that's what I'm primarily interested in.
You often hear about people discovering new organisms, but what about new metabolisms? New ways of processing carbon and nitrogen and sulfur. This is the kind of place where you can actually find something new in terms of metabolic diversity. Oh my gosh, that is a baby Riftia. Good gracious. Look at that. There's lots of food there, for sure. You just gotta be willing to, I mean, you're living literally inches from being boiled to death.
I love worms. People have been studying them for a long time. We're just continuing to do this with also a lot of new technology that has emerged in the past years. Yeah, they are very exciting systems, including us humans. We live with our gut microbiota and microbiota in our skin. The fact that we can study those dual relationships helps us also to understand really diverse microbiota.
When we think about maybe possibly finding life on other planets, kind of have to use your imagination of what that might be like. Chemosynthetic ecosystems are definitely a possibility.
So I saw this video and I just remembered that I actually have tube worms in my office, and I should have brought them out as props. It just completely slipped me. I only remembered when I showed the video because I certainly use it in class and I really should have brought it. But I hope that gave you a sense of how different hydrothermal vents really are. Not just from the perspective of microorganisms, but also from the perspective of the animals that inhabit these systems. But now we'll move on and talk about viruses. And when I'm talking about viruses now, from here on until the end of the talk, I'm primarily talking about viruses of microorganisms, not viruses of animals or any higher organisms. It's just viruses of bacteria. That's it. So to give you a sense and a little bit of perspective, how abundant are viruses in the oceans, or pretty much any ecosystem really? They are very abundant. If you look at, say, just the abundance here, so here are prokaryotes, so those are microorganisms, bacteria. There are protists, which are larger organisms, and say larger single-celled organisms, and then there are viruses. So viruses, about 95% of all biological entities in the oceans are viruses. That's how abundant they are. I'm calling them entities because there's a debate as to whether viruses constitute living things or not. But viruses constitute about 95% of all biological entities in the oceans. But if you look at, say, how much biomass they actually constitute, it's actually very little. So 95% by abundance, about 5% by biomass. That's because they are so tiny that they don't contain a lot of biomass. But because they are so numerous, they can be extremely important because they can continuously infect microorganisms, kill them, turn the entire ecosystem around. So They control the cycles of different nutrients. So how much iron is available, how much carbon is available, how much nitrogen is available in any ecosystem. It's for the most part, I would argue, dictated by viruses. They can influence microbial communities and the diversity of an ecosystem. And for the most part, we know very little about viruses that infect microbes in the deep oceans. So one interesting way to think about viruses, and traditionally how we've thought of viruses, and and some of the new aspects that we're beginning to appreciate about them. Traditionally, it was thought that viruses will go and infect an organism. So the traditional food web is that there are phytoplankton eaten by, say, larger organisms, grazers, and then a larger one comes in and eats them, and then an even larger one eats them, and that's your traditional food web. What does a virus do? A virus goes in, kills the phytoplankton. Now what does this organism eat? It can't eat these phytoplankton. What happens to the phytoplankton? It becomes, say, organic matter, or say dissolved organic matter, just dissolved carbon. Some other bacteria can consume it, and then this is a cycle that happens. But these larger organisms are not going to get to eat the bacteria. So this is something that we've begun to call a viral shunt. So rather than a traditional food web, what viruses would go in and do in an ecosystem is called short circuiting. So they're short circuiting a food web, and that's one of the most important contributions, let's say, of viruses that we've begun to appreciate in the recent past. Now, what exactly is the life cycle of a virus? How does a virus go about its business? Let's assume this is a bacterial cell. A virus goes in, and for the most part, viruses look like this. So depending on the kind of virus, you're looking at something like this, but they look pretty similar to this, is what I'd say. Of course, viruses come in a lot of different shapes, a lot of different sizes, a lot of genome sizes, but for simplicity, I think this is the best definition of what a virus would look like. So virus attaches to a bacterial cell, so it just goes and lands on it. And typically it can just land on it by random chance, because as I said, 95% of everything in the ocean are actually viruses. So they land on the microbial cell, and then they inject their genome. So they can actually inject it right into the cell. Once the genome goes in, the virus is essentially using gene expression and modulation of the bacterial cell. So it's getting the bacteria to make its proteins, its genome, and everything that it needs to assemble. So the virus then begins to assemble using all of the energy from the bacterium, resources from the bacterium, nutrients from the bacterium, and then eventually kills the bacteria and gets out of the cell. It's a very typical viral life cycle. Now, if you looked at, say, how viral growth or a viral infection would look like in a lab, you can grow microorganisms in a tube or anything in the lab, and it'll look something like this. So it's very cloudy. What that means is that there's a lot of bacteria that are growing in this tube. And over here, what we have done is we've infected the bacteria with a virus. So we've added a little bit of the virus in the system, and then we've just followed it over time. And as you can see, eventually it begins to clear up. So essentially, at a certain point of time, all of the bacteria have been killed. And there's probably, billions or trillions of bacteria in here. Each and every one of them has pretty much been eliminated by the viruses rapidly growing. So you'll see that this one is very clear. So it's pretty much, it's not exactly transparent, but it's close to that. It just means that the viruses have completely eliminated the bacteria. Now, for the most part, we study hydrothermal vents, but all of the processes that we study in hydrothermal vents don't happen in isolation. They also happen in other parts of the oceans. They happen at whale falls. Whales can be very large ecosystems when they die and settle down at the bottom of the ocean. They happen at these sites called cold seeps or gas hydrates. So these are also some that we sampled in the Gulf of California. So just like a hot hydrothermal vent, this is just some sort of a cold hydrothermal vent, but it primarily comes from methane, because there are these large areas in the seafloor where we have what we call methane hydrates that are being exposed due to increasing temperatures, and there's methane coming out. And methane is a very big greenhouse gas. And the other big ecosystem that my lab studies are oxygen minimum zones. What are oxygen minimum zones? So these aren't very well appreciated right now, but mark my words, in the coming decades, or even say 10 to 20 years from now, this is going to be one of the most important ecosystems on our planet. What is an oxygen minimum zone? As the name suggests, there are large areas in the oceans that have completely zero oxygen. Why? We don't have a really good understanding of the why right now. So there are a little bit of natural factors that play in, but what is absolutely certain is that microorganisms are also accelerating the expansion of these zones, and then increasing ocean temperatures are only adding to the problem. So there are these large zones in the ocean, no oxygen. So right now in the Gulf of California and the entire Eastern Pacific Ocean, so you go a little bit south of San Diego and then all the way down to, let's say, Chile, from 400 meters or say 300 meters to 1,000 meters depth, so a depth of 700 meters, this is no oxygen. So if you're a fish stuck in the surface ocean and let's say you want to dive into the depths, you're going to die. You can't breathe because there's just no oxygen in the system, completely zero. And these environments are quite similar to hydrothermal vents. So we're also studying hydrothermal vents and the processes that happen in here as a proxy for oxygen minimum zones. Now, how do you sample hydrothermal vents? We have to sample not just microorganisms but also viruses, and viruses are extremely difficult to sample because they're very small, so you have to filter very rapidly. So primarily use filtration, but we use what is called in situ filtration. So we have to build our own instruments, so I have collaborators around the globe who help me with engineering instruments and building these, and then we take them on robots down to the bottom of the ocean. So what you're seeing over here is an in situ filtration. This is actually called the super sampler. It can collect up to 24 water samples. We've also got some newer versions of this and a couple of other systems. But this has 24 filters, all of them, load one after another. They can get filtered at pressure at the depths of the ocean. It goes on to any robotic sampler. What you're seeing over here is one called Jason. And when the sampler comes up, we know, if you were in a hydrothermal vent or not. So, if you sampled something in the ocean, it'll look like this. It's white in color because it doesn't have a lot of particulate mass. But if you sample a hydrothermal vent, then it's just completely black in color. And the black color is coming from iron sulfide, so a form of sulfur and iron. When we sample, we load a sampler of any sort. It can collect fluids, it can collect viruses, let's say microbes. The sampler goes on a robotic vehicle, it goes down, and then we attach a hose. We stick the hose straight as close to the vent as possible and take it, because we can't stick it very close to the vent because the fluids that are coming out are about 300 degrees Celsius. So you have to be really careful where you sample. Microorganisms or any life does not exist at 300 degrees Celsius, but it exists up to 120 degrees Celsius. So we try and sample in that zone. So you really have to estimate where that zone is. And this gets trickier when you're in a submarine. Because when you're in a submarine and all you have is a window between yourself and a 300 or 400 degree hydrothermal vent, the pilot's trying to maneuver it, and he's like, tell me if that window is close and you're there. If it hits the window is going to melt, and so are you. But this is a tricky process of sampling and say where to sample. How do we actually detect a hydrothermal vent in the water, in that particular environment? So what you're seeing over here is a shimmer. So you can sample all of these different zones. So these are biofilms. So these are hydrogen sulfide consuming biofilms, all the white color over here. The waters are hydrothermal vent plumes, and you'll see a clear shimmer, and that's the hot water that's mixing into the oceans. And we can use sensors, and we do use sensors, but sometimes, you have to do things quickly. Your eyes are your best friend out there. So all of the colors are different biofilms. Here's a picture of a submarine, Alvin. So I was in Alvin in November. And Alvin gets launched off the boat, and it has 2 scientists and 1 pilot. So just the 3 of you. There are no restrooms on Alvin. So you're down there for 8 to 10 hours, and it's pretty cramped. It's a very small space. Alvin's about 1/3 of the stage, so that gives you a sense for how cramped it is. It is very cold because there's no heating in a submarine, so you just take 6 pairs of sweaters and maybe 2 blankets. But it is an experience of a lifetime, really. Alvin's also interesting. It's got people who, once you go into the water, they come up. There's actually a manual phone up there, and they ask you if everything's okay, if no water is entering, and if no water is entering, you can go down. But this is a picture that I really, really like. So this is Alvin, once it's come back up and we've sort of serviced the instruments. So yeah, that's Alvin. So it's got 5 windows. So that's the pilot's window. And then 1 scientist here, 1 scientist here. There's 2 more windows, but all we rely on are cameras. So there's a ton of cameras all around. Some of them, not great resolution. Some of them are pretty good resolution. But is there anything striking you see here in this? I mean, Alvin is a submarine. It's a multi-million dollar piece of equipment. But look at what the scientists are using. The scientists are using milk crates. So we are using, we have some bottles and some fancy equipment, but we have figured out that milk crates are actually fantastic. Nothing happens to them when you send them down to 3,000 meters. You send anything else, it comes back, it goes back this big, it comes back this big because of the pressure. But milk crates are fantastic. And they're very cheap, so we use milk crates for everything. A lot of instruments go inside the milk crates. It's just easy to use them. Here's another, this is not a submarine, so this is a remotely operated vehicle, but this is the most recent one that I used. This one's called ROV Sebastian. So this one's operated by the Schmidt Ocean Institute. ROVs and submarines are fundamentally different. Like the problem with submarines, one way to look at it is that they only give you about 6 to 8 hours of working time. So you go down there, you have humans on board, they need to eat, there's only so many things you can do, and there are no restrooms there, so you have to come back up in 8 hours. Actually, it's more because of the battery life, but still. Whereas with a remotely operated vehicle, it's connected to a tether, you send it down, it samples for you all day, all night, till whenever you run out of instruments or sample space or something like that. So the way they operate is fundamentally different, and both of them are fantastic. From a personal experience, going down in a submarine is some sort of a once in a lifetime experience, but ROVs are fantastic as well. Here is something we call a lander. This helps us a lot when we're out at sea. What this is just a really heavy piece of equipment that's connected to a float. So you send this down, and all this is giving you is basically some bench space. You need to store some sensors, some samples. So you send this down, it goes to the bottom of the ocean. You store samples on here. Again, this has got milk crates. So you stick your samples on here, and then you just pop an acoustic tag, and this is a float, so it just floats up to the surface. You go and pick it up. And then finally, this is the last piece of sampling equipment that I promised to show you. This is called a CTD rosette. So this is essentially 24 bottles. Each of these bottles is about 10 liters. You get some bigger ones too, but these ones are 10 liters, 24 bottles. You can send these down, and then there's a lot of sensors on here, and then whenever you're at a precise location from the ship deck itself, you can say that it should take a sample, and then it closes these tags and it picks a sample for you. So this is primarily for water samples, but it can take pretty precise samples. The Gulf of California was interesting with regard to using this particular equipment, because I just could not catch a break from getting jellyfish on the line. Like, I would just toss them and there'd be 2 jellyfish that would come up on every cast of mine. But these really are what I would call the workhorses of oceanography. We do a lot of work with these instruments because we're always bringing water up. So now, with regard to hydrothermal vents, let's try and understand what are the roles of microbes and viruses in transforming all of that hydrothermally derived sulfur that I spoke about. And this is work that in my lab is being performed by a PhD student, Christopher Kieft, who's also from Michigan. So he was also not happy on Saturday. And Chao Zhu, who's a postdoc in my lab. In our lab, we do a lot of DNA sequencing, and we use what we call a top-down approach to tracking microbial and viral ecology. What does this top-down approach mean? It just means that you go out, you collect samples, and what you're seeing over here is a filter. It's a filter that's collected hydrothermal water and has filtered it. So there are microbes and viruses on here. We extract all of the DNA and RNA from here, we sequence it, and then we try and understand three different sets of questions. We try and understand who is there in our samples. We try and understand what are the organisms that are there capable of doing. And then we try and understand how fast or slow these organisms are growing. All of this is done computationally. We can even extrapolate growth rates of the organisms computationally without ever growing them in the lab. We also do a lot of genomics. So everything that we do is genome resolved. And then we take these genomes, we reconstruct the precise metabolism of every organism and say the entire ecological role of a virus, and then we try and understand exactly what's happening, let's say, in an ecosystem. And the hydrothermal vent at the Gulf of California is going to look very different from the hydrothermal vent at Lao Basin. So that's why these ecological reconstructions are also important. But they come primarily from the metabolic reconstructions of every single organism, or nearly every single organism that actually lives in that environment. Now, when we think of viruses, it's actually pretty difficult to computationally recover viruses, because what does a virus really look like? We don't have a very good understanding of that. So this is something that we've really tried hard to understand, and this also involved us building our own software. So we've built something, this is built by my PhD student Christopher, and it's called Vibrant. And what this does is, given any complex mixture of DNA, it can actually extract and pinpoint which viral sequences they are and construct complete genomes and then tell you what the genomes are capable of doing. So it uses machine learning approaches to identify the viruses, and then once the genome's identified, it will characterize every single gene on that genome. And this is a really, really powerful tool for us, because microorganisms are difficult to culture in the lab. Viruses are even more difficult. So this is a really, really useful tool for us. So what have we learned about hydrothermal viruses? Remember, in the beginning I said we are most interested in how viruses actually manipulate the organisms, trick them into doing things they shouldn't be doing. That's some of the most important insights that we've recovered. So at hydrothermal vents, we have discovered these novel viruses that possess genes for oxidizing sulfur. Now, if you're wondering if you're reading this correct, yes, you are, because viruses should not be having any genes for sulfur oxidation or carbon or nitrogen or anything like that. Viruses are viruses, they go in, they kill the bacteria, they get out. No, but we've found viruses have genes, and we found not just one, but there are two genes on a lot of these viruses for sulfur oxidation. So if this is the entire pathway for oxidation of hydrogen sulfide to sulfate, this is what the viruses are doing. Or this is the step that the viruses are actually involved in. Why is it that the viruses are actually involved in this step? There's actually an interesting story behind this. So let's try and understand what the roles of such genes on viruses would be. And by such genes, I mean sulfur oxidation genes, or genes pretty much for any kind of metabolism. So viruses that infect these organisms, what they've done is they've acquired and retained parts of the sulfur oxidation enzymatic apparatus, and these kind of genes are actually called auxiliary metabolic genes, or AMGs. So AMG is what is the acronym that we'll use from here on in. So every time you hear AMG, it just stands for metabolic genes that the virus has actually stolen from the bacteria and then retained them on their hosts, on their own genomes, I'm sorry. So stolen from the bacteria and then retained on the genome, and then it's using it against the bacteria. So the viral genes, they don't evolve very rapidly. They evolve separately from the remaining viral genome, so they evolve very slowly. So they're very similar to the hosts. And what's most interesting is that they never represent an entire pathway like I just showed you. They just target something which, if you look at the ecology, it would be the weakest link in a chain, or say, it'll be a metabolic bottleneck of some sort. So evolutionary, viruses have just figured this out. So when we find genes like this on a virus, the first question we ask is, are the viral genes active? So what we do is we first produce computational evidence. So we produce computational evidence, we look at, say, protein folding predictions or a whole host of other approaches to check if these proteins are actually real, or are they just, remnants of something on a virus. Because viruses can have a lot of genes that just mean nothing. It's just difficult for us to even know what viruses are doing most of the time. But we've looked at a lot of things, including, perfect alignments. You look at the viral protein, you look at the bacterial protein, they're pretty much identical. So if the bacterial protein is active, there's no reason that the viral protein should also not be active. So then now what we've started to do is we've started to validate a lot of these AMGs biochemically. So we can do proteomics, so that looks to check if these genes actually form proteins. We look to see the activity during infection. So we have seen that a lot of these viruses will express genes specifically during infection, or say they increase it during the course of the infection. So there's a lot of interesting patterns that we've observed. And when we think of, say, how viral genes actually impact the bacteria, it really depends on the type of gene we're talking about. So the first one I'd like to briefly talk about is called DSR. And what DSR does is it's a gene that's specifically attacking intracellular sulfur globules. So there's one kind of virus that's stolen these DSR genes from their bacteria, and it's now helping the bacterium consume sulfur globules during infection. It's a pretty interesting mechanism, and I'll show you a model in a future slide. But what this bacteria does is it takes in hydrogen sulfide, and then it forms these intracellular sulfur globules. Now, why does it form these sulfur globules? Just like we store fat in our bodies as a food reserve, sulfur oxidizing bacteria store sulfur as globules, so that it's a food reserve for them. Something happens, they can go back to the reserve. So this is quite important to them. So because it's a reserve, bacteria always have access to it. So the viruses have retained just these genes, not any of the other genes in that entire pathway. So the virus infects the bacterium, forces the bacterium to use its intracellular sulfur globules. Different kind of virus has different genes. This one's called SOX, so that was called DSR. And in the SOX viruses, they actually force the organisms to generate intracellular sulfur globules. But it turns out they don't care about the intracellular sulfur. They're actually targeting a different compound, one that's called thiosulfate, highly soluble, present in the water. They're just forcing the bacterium to consume the thiosulfate that's around it, and the intracellular sulfur is just a byproduct. Different viruses, different strategies, pretty much the same outcome. Bacteria is dead, viruses are doing great. So let's look at a model of this virus-enhanced sulfur oxidation. So viruses will first steal genes for sulfur oxidation, and eventually this leads to uninhibited sulfur oxidation in infected host cells. And what happens with all of the energy that comes from either sulfur or thiosulfate is that all of the energy goes only to the viruses. The bacteria get nothing out of it. So before infection, you have a bacteria that is generating sulfur globules by oxidizing sulfur and then forming these intracellular sulfur globules. Bacteriophage or a virus comes in, infects this particular cell, and what happens? First, the virus would express the sulfur oxidation genes. As soon as they've expressed it, the bacteria are forced to upregulate their metabolism and forced into consuming all of their utilized stored sulfur. And all of the energy comes from this goes straight to the viruses. So, you can see the intracellular sulfur globules slowly decreasing in size, and then eventually there comes a breaking point and the cell dies. Now, this is one system. This is something that happens at hydrothermal vents. But one of the first things that we, after we observed this, one of the first questions we had as well, if it's being observed at hydrothermal vents, it's not happening in isolation. Okay, sulfur is there in our human gut, sulfur is there in Lake Mendota, it's there in pretty much every freshwater lake. what's happening in other systems. Can we find such viruses in the human microbiome? Can we find it in other ecosystems? So we were interested in looking at the diversity of these genes, looking at their distribution, how are they distributed throughout the environment and pretty much across the planet as well. And then finally understanding their ecology. Different viruses, do they carry different sulfur genes? What about genes for organosulfur? Because the human gut has a lot of organosulfur. Amino acids that contain sulfur, other compounds that contain sulfur, a lot of sulfur in the human gut, but bound to carbon. Now, when we first started looking at the oceans, it turns out that hydrothermal vents, clearly they're there, but we also found them in oxygen minimum zones. And initially, we'd only discovered two DSR genes and then 2 of these genes, but now we've found other genes as well. So clearly, if you find other genes, then the viruses are using slightly different strategies in the oceans. We've also found them in coastal zones, so surface waters as well as, say, coastal hypoxic zones. So these happen when there are these algal blooms in the ocean. And then right beneath them, we find microorganisms and viruses similar to what you see at hydrothermal vents. And we have also found them in other systems. So here are some genomes that we've recovered from other systems, very, very different. So this is Lactococcus. So this infects Lactococcus lactis, which also happens to be the state microbe of Wisconsin. So Lactococcus lactis is used to make cheese. So it's very, very important industrially. And viruses that infect Lactococcus lactis have these genes that degrade cysteine, which is a very important amino acid, and it produces H2S. Here is a virus that infects plants. We found, genes on this particular virus. Another one, plant virus, cholera patient. So an important virus that is, isolated from a cholera patient, and then finally from dairy. So, a phage that we found in dairy. So very, very different systems, all of them different genes, but all of these ones are actually involved with organosulfur. So either cysteine, the amino acid, or some other form of organosulfur. Now this is diversity with regard to environments. But what about diversity with regard to genes? So here is, all of the different pathways associated with sulfur transformations out there in the environment. So a whole load of pathways, different amino acids, different compounds. So these are all inorganic compounds out here. So sulfate going to sulfide, and then hydrogen sulfide and thiosulfate. And then the rest of it, whatever you see, is all organic sulfur. So your canonical amino acids like methionine, serine, aspartate, cysteine, as well as branch chain ones like, say, taurine. So it's all over the place. I mean, so many different genes associated with sulfur, and viruses have somehow figured out how to pick up all of these different genes, probably infecting many different bacteria, and each one of them is following some sort of a different ecological strategy. For the purpose of our human microbiome work, or say human gut, trying to understand human gut diseases and how viruses are associated with them, for the most part, we've been focused on cysteine and methionine, the 2 most important sulfur-containing amino acids, and how viruses are actually potentially accelerating the breakdown of these 2 amino acids. Cysteine specifically, because when cysteine gets broken down, it forms hydrogen sulfide, which is again a toxic gas. And we have found not 1 or 2, but 5 different kinds of genes that on viruses that can actually accelerate the breakdown of this particular amino acid. When you look at these viruses, how widely are they distributed? Pretty much all over the globe. Different kinds of environments, soils, non-marine saline, marine, hydrothermal vents, plants, fresh waters, pretty much everywhere. Every single environment that we've tried to get DNA sequences from, we've found viruses. When you think of, say, the human body itself, viral genes and viruses for degradation of organosulfur and production of hydrogen sulfide are quite abundant. You look at a random human, different kinds of microbiomes, be it the oral microbiome, the gut microbiome, the vaginal microbiome, you can find viruses that actually carry genes associated with sulfur metabolism of some sort. Now, I told you that we have focused specifically I mean, not specifically on cysteine, but we do focus a lot on cysteine. Why is that? It turns out that we've run a bunch of experiments associated with hydrogen sulfide that's actually produced from the cysteine, and it actually seems to increase viral fitness. What this means, what is viral fitness? What this means is that if hydrogen sulfide was present in an environment, then the viruses produce more progeny than if hydrogen sulfide was not present in the environment. So what this is showing is that, okay, there is a control, and this is the number of, this is the 100%, this 100% PFUs is a control standing for what you would get if there was no hydrogen sulfide. And then the moment you start adding hydrogen sulfide, so if you add 10 micromolar, you get about 150%. So 1.5 time increase in the number of viruses when there's this much sulfide, and then when you increase the concentrations further, it goes up all the way close to 300%. So 3 times the number of viruses. So as you increase the amount of hydrogen sulfide in the system, the number of viruses increases. And guess what? The viruses have probably figured this out as well. That's why they're retaining those genes and using them in the system. And what's important with this is that these H2S concentrations do not affect the microbial host. So it's entirely something that's happening for the viruses, doesn't impact the microbes. That's why it specifically has a link to viral fitness. So just like you saw for sulfur oxidation and hydrothermal vents, we've also developed a model for virus-enhanced sulfide production in the human gut. So before infection, you have bacteria that have a lot of intracellular cysteine. Cysteine, the amino acid, is extremely abundant, so millimolar concentrations inside a bacterial cell. Virus gets in, begins to express genes for degradation of this cysteine. The bacteria will upregulate their cysteine degradation, they produce the H2S, and H2S is great for the virus, so the virus replicates inside the host cell with increased fitness. All the cysteine eventually goes away, but so does the bacterium. And finally, the most interesting thing that I forgot to mention is that H2S gets out of the bacterial cell when the cell actually bursts open. And H2S in the human gut, gets out and the human gut is exposed to H2S. Now, H2S can cause intestinal inflammation, and that's considered to be, a precursor, let's say, to colitis or say what IBD, a bunch of diseases. So H2S is actually pretty bad, and H2S causes increased intestinal inflammation specifically. So if you're thinking of the healthy gut would not have any inflammation, it would not have any bacteria or viruses that actually promote inflammation. So healthy gut bacteria would have a lot of what we call commensal bacteria. So these are the good bacteria that are present in your gut. And then every time you have some sort of a dysbiosis, which means something's gone wrong in the gut and pathogenic bacteria have gotten in, you have this inflammation that happens in the gut. This can even cause some of the bacteria to get into your bloodstream, but there is an inflammatory response every time there are pathogenic bacteria. Now, what happens if you get a virus that can actually produce H2S? So a virus would actually increase the amount of inflammation in addition to whatever you're experiencing from pathogenic bacteria. And this is something that we have not quantified, we haven't understood, we didn't even know was possible, but we're just beginning to unravel. So with this, I'll just show you a brief summary of what I spoke about, but after this, I'll show you a couple of videos and just some cool pictures in general. But hopefully I've given you the sense today that sulfur metabolism is really important and dominates the ecology of hydrothermal systems. We've looked at a lot of viral metabolic genes that were involved in sulfur metabolism in different environments, including the human gut. And I've shown you that hydrogen sulfide increases viral fitness, increases the numbers of viruses. And finally, viruses really are a reservoir of diversity for sulfur metabolism genes. Now, I've spoken a lot about deep ocean viruses, so before I finish and before I show you some other pictures, I'd just like to show you a video of deep ocean viruses, what it takes to sample them, how we study them. I want people to associate viruses with the health of the oceans. Where there is life, there are viruses, and we can't see them. but sure enough, they're there, and the health of all of those animals, of all of those rich colors of biofilms really depends on the viruses. They are more abundant than all other biology by orders of magnitude. Much like large predators, viruses are just turning over the ecosystem and keeping things in check. Much of the research has really focused on viruses present in the surface oceans. It's microorganisms in the dark oceans critical to keeping oceans healthy, and it's these viruses that we know next to nothing about and that I'm here to study. I think hydrothermal plumes likely host one of the most diverse ecosystems, primarily coming from the presence of so many different chemicals in there. Back in 2014, we were part of a project where we discovered viruses that can actually manipulate bacterial hosts, and the way they do it is that the bacteria store sulfur inside their bodies as a food reserve. What the viruses would do is trick the bacteria into using that sulfur, and the bacteria keeps burning that sulfur, but the viruses derive all of that energy. And this is a very, very unique form of metabolism that's been observed nowhere other than hydrothermal vents. We have now taken a lot of these findings and are applying them to our understanding of human diseases, especially the human gastrointestinal tract that are associated with sulfur. And that would not have happened if we had not been down there studying hydrothermal vent viruses.
Unreal, right?
I think those mirror pools are a hotbed of viral activity. What's that underneath liquid? Well,
It's hydrothermal fluid. It's primordial juice fueling all of the biology that you see around the system.
the viruses we were going to find in here, we're not going to find anywhere else. Really, really unique, and I think it's really important to capture their diversity and understand their function. Think about the vastness of the ocean right in front of us. And all of this while, when we have been studying viruses, we have only been filtering small amounts of water. And that has its own sense of biases that come along with it. You think of, say, traditional virus filtration methods, it's about 5 milliliters a minute. So now you can see water coming in. Water is coming in through this pipe that's connected to these CTDs outside. So this is an inline filtration system. So what you're capturing over here are the largest microbes. The last filter is virus. And you can see how quickly it's going. This is designed to go somewhere around 1,000 times quicker than a traditional filter. So we can use these samples for genomics, metabolomics, all of this from just one set of samples. The first time that anyone's been able to achieve it at this magnitude, and we are immensely proud of this. The fact that we will be able to study microorganisms and viruses in their natural habitat here will help us learn about how they might respond to future global change. Sometimes the most powerful things cannot be seen, and I tend to associate viruses with that.
So before I finish, I think I'm running out of time, but I'll just show you a couple of videos. When we are down there, especially in a submarine, only one scientist has control of the cameras. I try and make sure I'm always that one, so that I can then take pictures like this. Here's a really nice hydrothermal crab. So I felt like I was having a staring contest with this guy. But very beautiful octopi, tube worms. So here is an interesting one where we saw this fish staring straight at us again. So some of these images, when you have the animals staring straight at you, these come out pretty beautifully. But this is a primary recreational activity on the boat. Ping pong. Especially on boats that rock. Some boats rock much more than others. It's pretty funny in which direction the ball goes, But we've also captured a lot of other interesting animals. Here's an octopus going after this lobster. A squid that has eggs in there. And then here's a really beautiful octopus. really big eyes. At one point in time, we could see the shine of the ROV in the eyes of the octopus. But there's also a dark side to do deep sea research in general. Increasingly, wherever we go, all we see is just garbage. All kinds. One of the most disturbing images that I've ever seen was a squid that had ingested some fishing line and choked on it and died. So you see the squid, and then you see this long fishing line come out of the squid's mouth. But you see garbage everywhere. And this garbage doesn't go away. Look at this. This is written in Spanish. I can't read Spanish, but if you did, you can see all the ingredients on here. This stuff doesn't degrade. It doesn't go away. You go down to the bottom, it's just littered. We had the opportunity to name a lot of hydrothermal vents on my most recent cruise. Most of them have pretty fancy names, except for one that was named garbage vent because it was surrounded by garbage, and we thought it would be, to get the attention of people, we purposely named it as such so that it gets out in the literature and people appreciate that all of these pristine, beautiful ecosystems are completely surrounded by garbage and nothing else. And before I finish, one of the most fun pictures I want to show is this one. So this is a hydrothermal vent, those mirror pools and just these hot vents shooting up. But we actually saw this funny image in here. It looks like a face. So zoomed out, and it looks like a face in here. Somehow this got captured in, and someone's eyes actually caught it. But before I finish, I would like to acknowledge a lot of my collaborators. I have collaborators all over the world for my deep sea and hydrothermal research, specifically Anna Louise, Chip Pryor, Simon Drew, Rika, Elizabeth, Meng, and Rupesh. And my other collaborators in human microbiome work, subsea floor microbiology and freshwater microbiology, and then finally my lab. These are the guys doing all of the heavy lifting in the lab and really keeping the lab going. So my PhD students, Chris and Patricia, my postdoc, Zishao, and Alyssa and Elise are about to join the lab, but they have rotated and spent some time. And then a lot of the undergraduates and my technician who really keep it going. And then a lot of this, what you saw, was funded by the Schmidt Ocean Institute. And then a lot of our sequencing now is funded by JGI, the Joint Genome Institute, and WRF has also funded a lot of my startup. So thanks to all the funders, and thanks to all my collaborators and the members of my lab. And with that, I will take any questions.
I'll start on that. Can you back up one slide, please?
Sure.
So the last name on there is Jill Banfield.
Yep.
And some of you may remember when she was here 20 years ago. And some of you may remember Cameron Thompson, who did the diving into the mines over by Platteville. And what's amazing to me about this is that this is all kind of related in the idea that you have things going on in water environments involving minerals, in your case sulfur, and that all this stuff is gets knitted or crocheted together eventually or not, but this is a pretty good example of something that you might not realize. And yet Tamara's work as a diver going and getting these samples goes back to Jill Banfield sending her down into these mines 20 years ago over by Platteville. That wasn't a question, but
Yes.
It's just a quick question. You started out talking about the ion concentration and gradient. You said sulfur metabolism or processing was important. You didn't touch on that, or did you? Or did I miss it?
I did not touch on that in the later part of the talk, but we do study dispersal and concentrations of sulfur, iron, and other elements in the oceans, both vertically as well as horizontally, but that's not something I covered. As far as iron is concerned, there are gradients. The closer you are to the vent, the higher the concentrations. Similarly with metabolism of iron, but as far as ion metabolism and viruses, so far we have not seen any indications. That's part of the reason we've sort of stayed away from ion specifically.
So I was just kind of trying to wait for how sulfur cycle, you said, was crucial to ion.
Correct.
Yeah, how is that connected?
So sulfur, so whenever you have any free ion in any system, ion can precipitate with a lot of compounds, but ion preferentially will bind to hydrogen sulfide. And what will happen is that if you have a lot of hydrogen sulfide in the system, ion binds to the hydrogen sulfide, precipitates out as iron sulfide. If iron sulfide forms, then ion's not going to get out into the surface oceans or say travel a lot, because once it precipitates, it just crashes down to the bottom of the ocean. So when microorganisms get in and they actually consume a lot of this hydrogen sulfide, they also give an opportunity to other microorganisms to form these extracellular polysaccharides that bind the ion, and that then allows the ion to actually go over very long distances, hundreds of thousands of kilometers. So it's a very complicated scheme of things there, but hydrogen sulfide metabolism by the microbes gives ion metabolism a chance. Yes.
How are you said there's some communities up in the plumes, but wouldn't they get swept down on those pretty fast, or do they have mobility to stay within those plumes?
So some organisms have mobility and some don't. Now, as far as plumes are concerned, plumes can be somewhat dynamic, but some areas of the plume are also very stable. So currents in general down there are not extremely dynamic, I would say. I mean, they are dynamic, but it's not really bad. So organisms have a reasonable amount of residence time in the plume. So a microbe gets swept into a plume, it slowly goes up into the plume, and then eventually, makes its way somewhere along with the gradient of the water. So it just stays in that environment for hours, I would say, 4 to 8 hours at least, I would say, roughly, give or take. But eventually those organisms, go to environments that are far away from the hydrothermal vent. The general understanding is that the deep ocean is pretty homogeneous, so you get similar or the exact same organisms being swept in all the time. Even though different individuals are coming into the plume every time, they're still very, very similar organisms because of the homogeneity of the deep oceans. Yes?
In the slide where you had the 4 test tubes with the bacteria that then went to clear with just the viruses, how long was that process to get to just the viruses?
So that particular one is a 2-hour infection cycle. So the first one was 0, then 30 minutes, then 60 minutes, and then about 90 to 100 minutes. So that's a 2-hour infection cycle, but it really depends on the virus. There are viruses out there that have 2-day infection cycles, 1-day infection cycle, or some that are really quick. So that was a Lactococcus virus, and that was 2 hours or less. It's just 2 hours and it's gone. Yeah, it's that quick. Tom. Okay, first.
I was just curious about the lactovirus in cheese making. Is it, do the ones that are used, the lactococcus used in cheese making, do they all have the virus? None of them have the virus? It changes if they have the virus?
So the lactococcus that are used in cheese making, they track the viruses very closely, because if viruses get into the system, they can just crash the entire system. So with viruses, I also did not touch. There are different kinds of viruses. So there are some viruses that actually integrate into the genome and just stay there, and they'll get out at some point in time. So these are called lysogenic viruses. So a lot of the lactococcuses have these kind of viruses. And when these viruses actually get out and actually begin an infection cycle and then kill the microorganism, we don't know. We don't really understand very well. So lactococcuses certainly have those viruses. They have multiple ones, the ones that are used in cheese making. But when you talk of, say, infection, or viruses that infect, so like lytic viruses, the ones we spoke about today for the most part, these viruses, when you're in an industrial setting, they take all sorts of precautions to make sure that these viruses never get in. All the water will be ultra filtered, because they never want to take a chance that their entire cheese stock will get, contaminated or say eliminated pretty much. Yes?
With bacteria that live in the deep oceans, you have to worry in sampling when you bring things up to the surface. How would you look for high pressure dependent viruses? What would that take from an engineering?
So that is a very good question. It's very difficult to bring microbes and viruses up without changing the system in some way. There is a threshold, and that threshold is about 2,500 meters. It's a loosely held understanding right now that if you're 2,500 meters or above, you don't need to worry about pressure as much as you would have to if you go deeper. With that being said, that's one of the reasons why we try and do in situ filtration. In situ filtration is great when you can do it, but you cannot do it in every setting. And if you do in situ filtration, you can't bring them up and then try and cultivate them. So it's a trick of, if you want to try and cultivate them, then we bring them up to the surface as soon as possible, and then we store them in a certain set of chemicals to ensure that we can bring them to the lab and then at least try and cultivate them. But if your only goal is to do DNA sequencing and you don't care about cultivating them, then you just filter them in situ or bring them up, just filter them. A lot of different ways. But that is a very good question, and unfortunately I don't have a very good answer. I mean, we recognize the problem, but we just don't have great solutions right now. Yes?
When you do culture them in the lab, are you using bacteria from those depths as the host, or do you have a substitute that you use as kind of a standard?
So viruses have a very limited host range. So they will only infect specific hosts. So if we are trying to get viruses from the deep ocean, then you have to infect them with bacteria from the deep ocean. There is just no other way to do it. So we are forced to use bacteria from the deep ocean. And in general, it really depends on some of the questions, but last semester I worked with the senior undergraduates in microbiology, and their project was to so I gave them a few of my samples, and they had to design media and cultivate them, and they cultivated 100 different isolates, 100 different bacteria. And our goal now is to try and expand this senior's project, senior honors work project, and this time, instead of say cultivating bacteria, use those bacteria as bait and then try and cultivate viruses. But that's some sort of the vision I have, with regard to student projects. So you need a lot of hands, and I think it's great for students to try and pursue this. Yeah.
Were they successful?
Yeah, they were very successful. 100. So there were about 50 students, so about 5 per group. So they worked in 10 groups, and each group isolated 10 different bacteria, and they didn't stop at that. They sequenced all of the bacterial genomes here at the Biotech Center, and yeah, they, seniors not just undertaking cultivation from the deep ocean, but then also sequencing the genomes and trying to understand what they can infer from the bacterial genomes from hydrothermal vents. It's a fantastic project, very successful, I'd say. Yes.
Changing the focus a little bit, you're studying very foreign environment to what we have here.
Right.
Thinking of a primordial kind of environment, are there any difference in prevalence of an amino acid encoded by the genome where you study versus what's on the surface?
Okay. So the short answer is no. Like, you'll still find all of the 20, 21 amino acids out there, but it's not a straightforward question because different organisms actually utilize them differently. So as an example, if you looked at the entire hydrothermal vent ecosystem, and the question was, in this ecosystem, will I find the same 20, 21 amino acids that I find in the surface oceans?
Or even prevalence of different?
The answer is you'll find all of them. but specific organisms evolve differently. So a lot of these sulfur organisms maybe are not, so they can produce cysteine, maybe others are not producing cysteine. So the patterns that you observe will likely be different in that environment, but you can certainly detect all of your basic 20 amino acids. Yeah, all of them are present. We don't see anything special on that front. Yes?
Do you expect your research to change, like with the changing climate and with increasing ocean temperatures? Does that matter the deeper you get?
It certainly matters because, so the deeper you get, the lesser you're going to see differences in temperature because the oceans are stratified. To give you a sense, the temperature in the deep varies between, I've seen between 1.5 to 4 degrees Celsius, depending on, say, where you are on Earth. So 1.5 Celsius, very cold, so that's about 34, 35 Fahrenheit. The surface oceans, the temperature changes are much more rapid. So I certainly see it impacting my research, but more for oxygen minimum zones and where we study the ocean water column. I also mentioned that we study Lake Tanganyika in Africa. Now that's entirely from the perspective of climate change. So I see it affecting my research, but not from a hydrothermal vent perspective specifically. Yes, Tom?
With the Lake Tanganyika example, I don't know of any place, first of all, I didn't know that existed as hydrothermal vents. We do have the Yellowstone vents. Do you know of anything that's deep at Yellowstone or any place in North America like that?
I don't know of any place in North America, to be honest. I actually have no idea how deep Yellowstone is. So yeah, I'm not aware. So Yellowstone's also a little different because it's my understanding that in Yellowstone, the entire system is pretty hot. So it's like a boiling lake. Whereas in Tanganyika, it's not a boiling lake. I mean, it's just a regular lake, but there is just some hydrothermal vents down there at the bottom. Tanganyika is also different. So there's just one lake that's deeper, that's Baikal. But Baikal is different because it's a temperate lake. So, it's cold, it's in Russia. Whereas Tanganyika is a temperate lake, is a tropical lake. And what that means is that tropical lakes actually are completely stratified. So Tanganyika, for just some perspective, is 1,500 meters deep, super deep. But there's only oxygen in the top 60, 65 meters. The rest of it is completely stratified. So no oxygen, completely saturated in hydrogen sulfide, methane, all sorts of toxic gases, and completely anaerobic metabolism. So all of the fish, the fishery industry, and everything that you can do with the lake, let's say, is only in the top 65 meters. The remaining 1,450 meters, just no oxygen. So that's the essence of Tanganyika.
My memory is 30 years ago there was a lake that belched. And can you talk about, does that bear on what you just mentioned?
Right. I forget which lake that was. That wasn't Kivu or, it was one of the African lakes, one of the tropical African lakes. But first I'll answer some of the previous question you asked, which is, are there other deep lakes? There are other deep lakes in Africa in that region. So there is Kivu, Malawi. They're all a few hundred meters deep, but none of them's 1.5 kilometers deep. With regard to the lake that turned over, belched, produced CO2, that is a very dangerous situation. I also happen to know that a little well because the scientist who discovered that was actually on my PhD committee. So what happened with this story is that there are lakes in Africa that have these geysers that produce CO2, carbon dioxide, and tropical lakes. And actually Lake Mendota as well. Lake Mendota in the summer is stratified. So what that means is the top of the lake is warm. It's nice and warm. And then the bottom of the lake is still cold. And because of this difference in temperature, there's a difference in gradient. So the top stays warm and that water stays here. The bottom stays here. They never mix. So the organisms and bacteria and viruses here never see these organisms. In Mendota, it only happens in the summer. tropical lakes year-round, they're stratified. So organisms in Tanganyika in the bottom never see the organisms in the top. They never mix with each other. And that is true in the lake that turned over as well. So the lake, so the part of the water that was at the bottom was super saturated in carbon dioxide. And just like, a can of Coke or Pepsi, anything under pressure holds a lot of gas. So when you're a deep lake and there's CO2 at the bottom, what happens? So it's super saturated in CO2. Something happened, okay, either temperature differences or say the lake that should not have mixed actually mixed. So the moment the lake mixed, all of that pent up pressure of CO2 was just released overnight. And it so happened that when CO2 gets released, carbon dioxide is heavier than air. So the CO2 got out of the lake and carbon dioxide is actually heavier than air. So what happened? All of the air went up, the carbon dioxide settled down. So imagine if all we had to breathe here was carbon dioxide, what would happen? We couldn't breathe it. So entire villages were wiped out overnight. People just died in their sleep because they didn't know what hit them. Livestock lost, entire villages wiped out. Because, everything in the surrounding of these lakes. Yeah.
But there was no smell.
No smell.
The plants did fine, and that was one of the, when it first came out, it was like, what is this? Because people thought it might have been hydrogen sulfide, but that should have smelled bad.
Yeah, hydrogen sulfide should have smelled bad. There would have been clues with regard to certain minerals turning blacker, impacting the plants, but CO2 did not impact the plants. There was no smell. Hydrogen sulfide has the smell of rotten eggs. You can tell that from a mile apart. But CO2, no smell, completely odorless, completely colorless. Yeah, just really bad.
Who was that person who was on your committee?
George Kling. So he, along with another USGS scientist that I forget who he was, they were the ones who were out there. Yeah, they've done a bunch of these BBC documentaries and stuff about these lakes.
Any other questions? One more?
This is a very narrow question, but how do the viruses sequester the energy when they are hijacking the bacteria? How do they ensure they get the energy enough?
So in theory, the viruses themselves are not sequestering the energy. It's just the bacteria that is redirecting its entire metabolic output towards the viruses. So when I say the viruses are sequestering the energy, all it means is that all of the energy has gone into building the viruses. So for example, building the viral envelopes, creating viral DNA. So sequestering the energy just means all of the energy is consumed by the bacteria to produce matter of some sort for the viruses. So the viruses are just benefiting from it. But strictly speaking, the viruses cannot do anything. It's the bacteria being manipulated to do it for the virus.
Thank you very much.